function we identified, surface epithelia and glandular. Main function of surface epithelia to form barrier between exterior and interior. Glandular epithelia function is secretion. Surface epithelia, according to relation to basement membrane, we divide into the simple and stratified. And today, last class, we consider simple epithelium. Today, we should consider stratified epithelium and glandular epithelium. Here, you can see diagrams of stratified epithelium. Among stratified, we can identify keratinized, non-keratinized, and transitional. Keratinized epithelium consists of the 5 layers. Not this mistake. This keratinized, this non-keratinized. Keratinized and transitional consist of these 3 layers. I remind you that stratified epithelium have only 1 layer of cells that lay on the basement membrane. Other cell never contact with basement membrane. If we examine some photos, we can see stratified epithelium. Where is your white coat? Look please at this photo. This is stratified keratinized epithelium where we can see 5 layers. First layer, basal, that lay on the basement membrane. Next layer, what all this, spinosal. It consists of several layers of cell. Next, the darkest layer will be granular layer. Above it, very narrow, here it looks like red color, lucidal, and after that corneal. Lucidal and corneal consist of dead cell. They lose the nuclear, so these particles what left of the cell. And only 3 first layers have alive cells. Such type of epithelium we can find skin of palm and soles. Another type of the this so named thick skin because consists of the 5 layers. Other surface of our body cover. Please leave the class. Leave the class. When you put on white coat, you can come in. Close the door. Thin skin. Так, сейчас посмотрим сколько. You are not permitted for the class because you violate more than 1 hour. So it's my right. It's my right. Так, все. Your class begin at what time? Все. Both you So, as the surface of our body cover with so-named thin skin, in such case, what stratum lucidum, red color, will be absent and corneal layer will be very thin. Such skin, epidermis of skin, has hair follicle. Let's look at this epithelium. It's also stratified, which consists of 3 layers. First layer, basal. After that, spinosal, and look please, superficially situated cell, superficially situated will be flattened. This is very important to memorize, because if we examine this type of epithelium that we name as transitional, it also consists of 3 layers. And first layer, basal, that lay on the basement membrane. Next layer, intermediate, we can see nuclear cell that situated above basal. And superficially situated cell, look please, have round, large shape. Compare with this flattened cell. This is main difference between transitional epithelium and non-keratinized. Non-keratinized epithelium we can find in oral cavity, in cornea, for example, in esophagus. Transitional epithelium can be found in urinary system. This diagram of this type epithelium. And if you remember, all epithelium have morphological features. So position of this epithelium will be on the body between exterior and interior. Cells form layer using intercellular junction. Epithelia rest on the basement membrane, and this basement membrane initiate polarity. In case of this simple epithelium, polarity, signs of polarity, we can see in specification of apical and basal surface. In case of the stratified epithelium, we can see difference in layers. For example, if we compare basal layer and superficially situated cell, they look differently, yes?
The 6th one is the keratinized layer, right?
Yes, this keratinized layer, corneal. In case of the non-keratinized, here we can see flattened cell, this cuboidal. In case of the transitional epithelium, this small cuboidal, this large round cells. Here we can see part of the epithelium, stratified, keratinized. Basal layer, spinosal. Here we can see several layers of cell with granules inside. This is granular layer, this lucidal and corneal. This is process of keratinization you can read later. Now let's examine more more slides of this type of epithelium. For example, what you can see, small magnification, stratified keratinized epithelium. So basal layer, spinosal layer, granular layer. Granular will be the darkest, lucidal and corneal. In your bottom book, task 31, you should draw piece of the stratified keratinized epithelium here. Not pay attention to this diagram. This is look like the electron micrographs. So no, you are late. You are late more than so.
I don't say.
Don't disturb me. Respect your classmates, please. I not allow you to enter.
Okay.
I not allow you enter. So you are late. So you. They come, you not. So please close the door.
Professor, a stratified keratinized bacteria.
So you should draw here all layers of stratified keratinized epithelium. I explain how to look correctly.
A stratified keratinized bacteria.
So, well, for example, this is basement membrane, and first we draw cuboidal cell. When we draw this cell, you should remember that cell form layers without any interaction. That's draw, I use previous wall of the cell and add new cell, yes? This is very long, I will be short because it will be a lot of time. Next cells I add to previous. This cell belong to basal layer. This spinosal layer. This cell granular layer. This cell the darkest. That's why I add color to them, and in the cytoplasm we can add granules. Granular layer, and after that this cell dead and form particles that joined with each other by material of granules. This granule we name as keratinosomes. They sleep with each other all this mass of the cell. That's why we know we don't draw here cells, these only particles, and this is lucidal, and this, the thickest layer, will be corneal. You can be free.
Sir, professor.
You can be free.
Tunnel să cu tot asta, classes cancel.
Sure, you can be free.
Mal puțin.
Da. Let's look again at the photos. Basal. After that, spinosal. The darkest layer, granular, lucidal, corneal. Yes? Look like my drawing. So, you should draw in your task 31 approximately such picture. For example, one more. This is basal layer. This is spinosal, the darkest granular, lucidal, corneal. High magnification. Look, please. We can see in case of the granular layer very clear these granules. This is spinosal. This basal layer. This is lucidal and corneal. Okay. Also, I would like to return to this diagram. This diagram, the same time epithelial, but among epithelial cell, we can see another cell that belong to different differons. For example, pigmental cell. Who remembers source of origin of pigmental cell? After embryology studying Who remember pigmental cell arise from neural crest. As Merkel cell, this sensitive cell belong to nervous tissue. They also arise from nervous neural crest. Langerhans cell. This is cell that we belong to macrophages. So they arise from blood stem cell. So among keratinocytes we can see another difference. And what this diagram here on the left demonstrate for you these different difference. And here you should identify these cells. Where is pigmental? Where is Langerhans? Where is Merkel cell? You should identify only here, not here. This you only draw layers correctly. Okay? Sorry. Next type, epithelial. Stratified, non-keratinized. So it consists of the 3 layers. There are no precise body between layers. Layer transformed to next layer approximately. So we can divide this, we identify layers in non-keratinized epithelium only approximately. No, these cells that belong to basement membrane here, we name as basal layer. After that, what all this cell, we classify that cells that belong to spinosal layer. And when we can see that cell became flat, we name this layer as squamous layer. So, on the drawing, again, cells that lay on the weight membrane, basal layer. After that, spinosal. But then they became more and more flamed. Yes? Nuclear repeat shape of the cell. They became more horizontal, oval. This more and more flamed. So approximately here spinosal. And this, where we can see only flamed cell, this squamous layer. So let's examine slide with this type. As I said, we can see this type of epithelium in esophagus. Esophagus, this is tube, and internally internal surface of the tube lined by this epithelium. What here? What this? This epithelium. This tongue. Esophagus. Okay, esophagus. Again, and where is just maybe let's look at this slide. So we can see here very clear. We can't see cell membrane, but we can see nuclear. The shape of the nuclear informs us about the shape of the cell. So we can see what basal layer, where we can see oval and round nuclear, this spinosal layer, and where we can see flattened nuclear, this is squamous layer. Is it clear? Yes. Okay. Next type epithelium.
Because the nucleus is most likely in the shape of the cell that is in it, so when the cell is...
nuclear, repeat shape of the cell always. Next type epithelium. This is transitional epithelium. Such type of epithelium we can find in urinary system, for example, in urinary bladder. Such structure of epithelium correspond to function of urinary bladder. It store urine, and wall of the urinary bladder can stretch. Yes. So epithelium should also undergoes by stretching. That's why, look please, we can see basal layer, intermediate layer, and large round cells on superficial layer. And when wall of the urinary bladder stretch, epithelium, this cell became more flattened, but after that return to initial state. So if we compare this and this, again we can see basal, layer. After that, cells with round nuclear. This layer we identify as intermediate. And this cell, superficially situated, will be very large and round. This superficial. So, main difference between non-keratinized and transitional shape cells of superficially situated layer. Yes? Here we can see flattened, this round, large. This is one more. Look please, round nuclear, round cell. One round nuclear, round cell.
But they change their shape when they're stretching, right?
Yes. There is just one diagrams. Look please, what, for example, here you can see two functional states. This is usual, this in case of stretching. So what two photos correspond to this functional state of epithelium. This is all with stratified epithelium. Now our task to consider glandular epithelium. And of course that task number 13. This is stratified, non-keratinized. You should draw as here.
Okay.
As here.
Okay.
Task 32. You have read diagrams of transitional epithelium. You should only color it and name our necessary structures. Yes,
Okay.
layers, and so on. Now, glandular epithelium. Main function of glandular epithelium. This is secretion. They secrete some substances, yes? And first, let's consider some general terms. In our organism, We can classify glandular cells that produce some chemical substances according to chemical nature of these substances. If cell produce protein, it look like this. If it's produce mucus, it look like this. That's why we can identify protein secreting cell or serous, mucus secreting cell. And cells that secrete water and salt, this is glands in the skin,
Or sweat.
the sweat gland, yes, and glands that produce steroid or sebaceous, lipid nature. Yes, for example, this is sebaceous gland of the skin. And let's compare these two cells, because our task to study, to differentiate this type, because these cells we can see a lot. Protein secreting cell more tend to be cuboidal. We can see colored cytoplasm, and that's why round nuclei. So I choose three criteria: color of cytoplasm, shape of nuclei, position of the nuclei. Position of the nuclei in the center of cell. Let's look at this mucus. Cytoplasm very pale or sometimes unstained, because these granules that contain carbohydrates not well stained with our dyes. Look please at the shape of the nuclei. It's flattened, and position of the nuclei. It's not in the middle. It's move more close to basal surface of the cell. So three criteria that help us to identify where is protein secreting cell, where is mucus. Color of the cytoplasm, shape of the nuclei, and position of the nuclei. And it does not matter what type of the secreting glandular cell, but in any glandular cell we can see several phases, and these phases we name as secretory cycle. First, cell uptakes chemical precursor and use it for synthesis using rough endoplasmic reticulum and Golgi apparatus. After that, it accumulates in apical surface of cytoplasm, and next phase it released outside by exocytosis. Сейчас one more. And releasing secretion can be different types. Typical exocytosis we name as merocrine modes of secretion, merocrine type of releasing of secretion. But some cells release secretion with particularly destroyed with part of the cytoplasm. Such type of releasing we name as apocrine. And holocrine type secretion when cell dies, and after that released all accumulating granules. Give example of such type gland, because in such type, if cell dead, that they should have a new cell. That's why such a gland usually consists of several layers. Here only one layer, this more than one. Потому что да. What is it? This is class number 4. What is it?
Sebaceous
Sebaceous gland, yes. What sebaceous gland release secretion using holocrine type. Cells destroyed and released. Sweet gland can have merocrine type secretion and epocrine. This is in general. Now let's return to classification. In our organism, we can see two type glands, endocrine and exocrine. Main difference between them, exocrine glands release secretion into the surface. That's why they have epithelial, they have duct for that. So they have cells that secrete. After that, to secrete this outside on any surface, they need duct. So two parts, secretory part, duct. Another type of the glands that released secretion into the bloodstream, into the blood vessel. Such glands we classify as endocrine, and there are not necessary for them to have duct. That's why main difference between exocrine and endocrine, this is presence or absent duct. For example, what here we can see, let's compare this cell and this cell. This is exocrine or endocrine, and this, we can see here granules and these granules. Which of these cells will be exocrine?
They have duct, so...
No. Now we compare only the cells, two cells. Among epithelial cell, we can see that, for example, this cell and this. Let's compare. Which of these cells will be exocrine?
Upper
Number
one is
two or
exocrine
upper?
and number 2 is endocrine.
Number 2 is exocrine.
Okay.
Because this exterior for the cell. Yes, they secrete this in exterior, in some space. Here, granules accumulate in basal part of the cell. That's why they will be released in underlying blood vessels. So these endocrine cells. What we can see endocrine cells here, accumulation granule in basal part. And in case of the last class, when we consider simple epithelium, we can see goblet cells. Yes? Goblet cell will be exocrine or endocrine?
Goblet cell.
Where they release secretion into the bloodstream or into the some space. So
Because, it has in the apical part, so it's
apical part, so
it's exo.
Exo, yes. Outside, in surface. What this is, for example, intestine, yes? Intestine tube and internal space. This is external space for our body, yes? Because food and digestive tube begins from oral cavity, and that's why this space connect with air, for example, yes? So this exterior for our body. And epithelium that's situated here released secretion to exterior. So goblet cell will be exocrine cells.
For example, in epithelium columnar, we have the goblet cell.
The columnar, yes, look please. If you did your homework to this class, no such question you have. For example, among epithelial cell, we can see goblet cell. Goblet cell that accumulate mucus granules. That's why they look like holes. What here? Goblet cell among epithelia. So in such case, gland correspond to one cell. One cell, one gland. But we have multicellular gland in our organism. In such case, there are no place for this cell among epithelial layers, and we can see multicellular glands. They situated internally, but they connect with the surface, outer surface. This is secretory portion, this is duct, and secretion will be through the duct again on the surface. Yes? So, in our organism, we can find unicellular duct, goblet cell, and multicellular glands. As here, look please, we can see these glands, secretory part, but the duct will be open here. Just, and that's why such multicellular gland, which consists of the duct and secretory part, we divide, we classify according to
Branches of
duct structure and secretory portion structure. So, duct can be unbranched and branched. If it's unbranched, as here, we classify such gland as simple. If we can see a lot of branch, it's divided into the branch, we classify such gland as compound. After that, secretory portion can be unbranched or branched. Shape of secretory portion also can be different. Alveolar or round as here, or tubular, what such flattened. So, full classification of glands can be simple branched alveolar or simple unbranched tubular. Compound glands always have branched secretory portion, and again shape can be different. Let's try. Give classification to this gland. If it's
Simple.
duct and this, yes, simple because duct unbranched. After that, branched because we can see several secretory portion. And what shape of this? Tubular. Simple branched tubular. Is it clear? Let's give classification to this. Compound, branched, tubular vesicle. Yes? Because we can see as tubular as vesicle, secretory portions.
tubular vesicle is the tubular alveolar, right?
Yes, it's the same, synonyms. Well, again, you can train yourself. This is photos of switch gland with duct. This duct open on the surface of the skin. This hair follicle, hair follicle, and sebaceous gland open to hair follicle. Look please, this sebaceous gland, that open to hair follicle. This is main characteristic differences between serous and mucus. We can see a lot, but remember about three criteria: color of the cytoplasm, shape of the nucleus, position of the nucleus. Using this criterion, you can identify very serous and mucus without any difficulties. Let's look. What, for example, we can see secretory portion, which consists of serous cells that produce protein. So, cytoplasm of this cell well stained. They have round nuclei. in the middle or in the basal part. This is mucus cell, which consists of cell which cytoplasm unstained or very pale stained because mucus contention, flattened shape of the nuclear, and in basal part of this cell. Так. So, in task number 47, you should draw mucus secretory portion and serous secretory portion. What necessary to do? Cross section, two cross sections through the secretory portion. For example, this will be serous, this mucus. I draw serous cell. They tend to be more cuboidal. Their cytoplasm well stained, so they darker. Round nuclear, approximately in the center. Mucus more columnar. Unstained and nuclear flattened in basal part. It's enough, and you can identify these without any difficult. Let's do it. For example, let's look at the tongue. This is tongue. Tongue covered with what type of epithelium? Here. Simple or stratified?
Stratified.
Stratified. It consists of 5 or 3 layers?
3.
3. So 3 layers we know non-keratinized and transitional. Look please at the shape of the cell. They large, round or small flattened?
They're not keratinized.
Non-keratinized.
Non-keratinized.
Non-keratinized. So what such?
Professor, I want to say something.
Так, after class, please.
Okay. But if we are outside because of our leader just did
Так, after class, not interrupt my explanation. Respect your classmates. So, first you should decide it's simple or stratified. After that, you should decide it consists of 5 or 3 layers. And if it's only 3 layers, so you should identify this transitional or non-keratinized. That's why it's easier to identify epithelium. Now look please in the thickness of the tongue. We can see 2 type glands. This is dark, well stained. This is pale enough. Yes, a lot of secretory portion. So tell me please, will be serous or mucus secreting cell?
Serous, because
Serous. And this? Mucus. Now let's examine again. Serous, mucus. Serous, mucus. High magnification of secretory portion of what? Serous.
Yes.
Serous, yes. Again, serous, round nuclear we can see here. And this?
Mucus.
Mucus.
Pale,
Okay.
it's pale cytoplasm and nucleus is on the basal.
Так. And also we examine for the task 34, we examine simple unbridged tubular gland. Such gland we can find in the wall of the head. This is uterus, lumen of the uterus, and we can see a lot of glands that open here. What, for example, here, lumen of the uterus, this is gland. This is secretory because they have tubular shape. It's difficult to identify where is duct, where is secretory portion. No, approximately what this very narrow part will be duct, this secretory portion. And you should draw one such tubular, simple tubular gland in task 74. So, we intend to leave for example, this is basement membrane I draw, and this is epithelium that like lumen of the uterus, and this again epithelium that form gland. And lumen of the gland will be very narrow. What is lumen? And in such gland, we can identify different parts. These we name as neck, body, and bottom. Neck usually will be duct. Body and bottom, this is secretory portion. So when you draw, here will be underlying connective tissue. Yes, so this lumen of the uterus, this lumen of the duct. You should draw approximately such picture. One more task that necessary to do, task 36. You should draw another gland which is simple, branched, and alveolar. And such gland will be sebaceous gland. The second time when we meet sebaceous gland. For example, look at this hair follicle, and on the line that divides first third of the hair follicle, we can see gland, and this gland consists of several lobules. Usually, here we can see two. For example, here 1, 2, 3. So secretory portion will be branched. Yes. And shape of secretory portion tend to be alveolar. That's why it has one duct but several secretory portions. This is epithelium, and you remember that sebaceous gland, stratified gland, because it has holocrine type secretion, yes? So here also should be full of the cell. No, I will be my research. And some of the cell, if you remember, form undifferentiated cell, after that differentiated, after that cell with karyokinesis. Yes? So I think you drew it correctly. But today we pay attention to demonstrate that it's simple, and alveolar gland. Так. So, task 33. This is
Goblet.
cycle of the cell. So, actually, you can use these phases. You should name these phases secretory cycles. Task 35. You can see different type of glands, and your task to classify each of them. That's all. And 38. You can see table, and this table in relation to classification of gland. First, how we classify glands according to duct, simple compound, yes. After that, according to shape of secretory portion, tubular, alveolar, da? This is classification according to mode of secretion, holocrine, merocrine, and this classification according to chemical composition of secretory granules. So, protein, serous cell, mucus, water, salt, lipids produce, yes, all this. Is it clear what necessary to do? Because next our Thursday is colloquium. To colloquium, you should have your working book signed. So, if you don't miss class, you should find me and I ask you, and if you answer, I sign. If you missed class, you should go to отработка and try to sign this class on отработка. For отработка, you should have all tasks made. You should orientate in material and answer the question of the teacher on duty, on отработка. Отработка for you on Monday, Tuesday and Thursday at 5, auditorium number 5. But this auditorium number 2, further in corridor, you find number 5. So not be late, because now our отработка not so many Russian students, that's why it's lasts fast enough.
Professor, if we have just one отработка, can we do the colloquium?
It does not matter how. You should have all your classes signed.
Okay.
So you